So the name for the mouth is also called the buccal cavity. It is formed by cheeks, lips, and palate. It encloses the teeth, tongue, and salivary glands. So the most important activities happening in the mouth is saliva production, which is secreted by the salivary glands. So this one is the problem, right? Yes. Okay, this is some anatomy that you need to appreciate regarding the buccal cavity. This is the uvula, the tongue, okay, and other structures that you are very familiar with. Okay. The salivary glands, you cannot see them here unless I show you one of these diagrams. But there are minor salivary glands around the buccal cavity. Minor salivary glands. Some of the minor salivary glands, you find them on the lips, on your tongue, under the tongue, the cheeks. Those are minor salivary glands. But the major salivary glands, there are three of them. Which are what? Parotid gland, sublingual glands, and submandibular. Those are the three major salivary glands. So other minor glands are just located all over the mouth. We will not discuss those, but they produce saliva. Straightforward functions of the mouth. Ingestion of food materials, chewing the food and mixing it with saliva, appreciation of taste, transfer of food to the esophagus by swallowing, plays a role in speech, social functions such as smiling and other expressions. Those are performed by the mouth. Appreciation of taste, because on the tongue there are taste buds. Under special senses, when you do neuroscience, neurophysiology, you do the special senses. Neurophysiology, you've heard of neurophysiology, third year. Yeah, somewhere there in third year. So that's a type of physiology we do in third year. Neurophysiology. Oh yeah. You look at all these special senses, the ears, the eyes, taste sensations, the motor system, pain, how pain is produced. Okay, so appreciation of taste of food done by the tongue, chewing of the food and mixing it with saliva, that is mastication. Ingestion of food materials, and raw in speech. So these are the three major salivary glands. We have the submaxillary gland, sublingual gland, and parotid gland. So the parotid gland is the largest. Okay. So those are the major glands: parotid, sub, maxillary, and sublingual, under the tongue. Lingua meaning under the tongue. And you find that each gland is associated with a duct. For example, the parotid gland, the duct is there, it's called the dispensing. for the submaxillary gland, the duct is called the Wharton duct. For the sublingual, it's not shown here. Okay, so these ducts, they transport the salivary secretion. These glands are classified into three types. Salivary glands are classified into three types based on the types of secretions: serous, mucus, and mixed. Serous, mucus, and mixed. So serous glands are those that secrete what? Watery saliva. Those are called serous glands. They secrete watery saliva. And mucus, just from the word mucus, thick. These glands, they secrete thick saliva. And then mixed glands, they're able to secrete both types, serous and mucus. So from the notes, what you can see that which glands are serous. When you're reading down here, when you're reading this slide, down here I put notes, right? I'm sure you've seen them before. Yes. Now as we are reading the slide. So serous glands, it's a parotid gland. Mucus glands, these are usually minor cells, minor glands, and then mixed glands, we have the sublingual and submandibular. Okay, so the gland that secretes serous type of saliva, watery, okay, it's thin. Those are called, an example is the parotid. Mucus glands is mainly secreted by minor glands. And then the mixed glands, they secrete both mucus and serous. These are the sublingual and submandibular glands. So saliva is, from the physiology, a normal human being, the properties of saliva is as follows. Volume, it is 100 or 1,000 mils to 1,500 mils. It's secreted per day. Okay, a normal healthy person at least, on a daily basis, 1,000 to 1,500 mils. Reaction, the pH of saliva is 6.35 to 6.85. So it's slightly acidic. Slightly acidic. Because 7 is a neutral. These fluids, we should be able to know their pH. For example, gastric juice is 2. Saliva is about 6.85, slightly acidic. Pancreatic juice is alkaline due to the bicarbonates. So it's usually above 8. Okay, so for this saliva, it is acidic. Specific gravity, it ranges between 1 to 1.002 and 1.0112. Don't feel lazy to remember these decimals. Very important. 1.002 and 1.012. come? Are you sure amylase works in an alkaline environment? But how come it also works? Salivary amylase. Because there are two types of amylase. There is salivary amylase and pancreatic amylase. Pancreatic amylase is released from the pancreas. So maybe you mean the pancreatic amylase from the pancreas. That one, yes, it works in alkaline environment. That's what you mean, I think. Actually, this is the one which is very significant when it comes to digestion of carbohydrates, the pancreatic amylase. Yes, in terms of significance in digestion of carbohydrates, the pancreatic amylase. That's why you find the amylase, after you swallow your food, it goes into the stomach. The salivary amylase is not active in the stomach because of the pH. Yes. Okay, so take note, there are two types. There's the amylase usually we refer to, it's the one from the pancreas, which does a lot of digestion of carbohydrates and starch. It's clear? Specific gravity, when you compare the density of saliva to water, saliva is more on the thicker side. Tonicity, I don't know if you can see this, tonicity is number 4, there is tonicity. Saliva is hypotonic to plasma. Yeah, you're able to see this point? Yes. Okay. Saliva is hypotonic to plasma. Hypotonic, what does it mean? Forgotten in biology, hypotonicity, hypertonic solution. Yes. More water as compared to plasma. Yes. So saliva is hypotonic. It's more diluted as compared to plasma. We're going to see the reason why it is hypotonic. When it is being produced, you're going to see the reason why it is hypotonic. It has less sodium, it has less chlorides. But for plasma, the chloride levels are high. Okay, so let's remember these properties of saliva. So if you have a patient with hypersalivation, it means that saliva levels or secretion are above 1,500 milliliters per day in 24 hours. Hyposalivation, it means they are low. Okay, about 1,000 mils in a day. If the saliva is too acidic, below 6.35, it means there's something wrong. Okay, could be acidosis, and many other pathological conditions. Specific gravity saliva should be that. Tonicity saliva should always be hypotonic to plasma. So that's the properties and composition of saliva. So what is in saliva? There are a lot of things in saliva, but these substances can be classified as either water or solids. Water, very important, 99.5% it's water. Solid is just 0.5%. So among these solids, it can further be divided into organic substances, inorganic substances, and gases. We know what organic substances, these are carbon-containing compounds. Those are called organic substances. Inorganic substances are sodium, calcium, these electrolytes. Sodium, calcium, potassium, bicarbonate, bromide, chloride, fluoride, phosphate. And in saliva, there's some gases dissolved, which is oxygen, carbon dioxide, and nitrogen. So it's one of the important, for laboratory investigation, it's one of the samples that can be gotten from the body. That can be obtained from the body. You want to do your analysis. Saliva, blood, stool. Okay. These can be used for analysis purposes. Normally, there's an important statement here. Normally, glucose is absent in saliva, but if it is found in conditions such as diabetes mellitus. Diabetes mellitus, it's a metabolic syndrome where the metabolic processes have gone wrong. And it presents with hyperglycemia, high levels of glucose in the blood. So when it is found in saliva, when glucose is absent, it's usually absent. In a normal healthy person, it is absent. But when it is found in saliva, it can mean a lot of things. One of them is diabetes mellitus. Okay, these are other, when we go into detail now, the various composition of saliva. Most of the things found in saliva usually are found also in blood, right? Saliva is black, actually. Yes. Saliva is black, so don't play around with the people's, saliva. Don't play around with saliva, because just look at this composition. It has proteins, epidermal growth factors. And we know proteins, they are segments of DNA. Buffering. So when you're in a lab, you're analyzing these samples, make sure you just wear those gloves, right? Don't you do experiments? Do you do labs nowadays? Not nowadays, in physiology. Next week. Next year. Or you are starting today. So in case you are dealing with samples to do with the saliva, saliva can be very infectious because it's just a component from blood. It's made from blood, actually, plasma. So don't take it for granted. Make sure that you take precautions. It can be very infectious, just like blood. Lubrication, mucins, all these are composition of saliva. So this is the structural and functioning unit of the salivary gland. Are you sure I didn't do this? Structural structural and function unit of salivary gland. We said the salivary glands, they look like a bunch of grapes. So when you pick up one grape, this is the structure, that's how it looks. This is a structural and functional unit of the salivary gland. The unit that actually produces the saliva. So what are the parts? We can start from here. Of course, saliva should be produced from the cells. It should be stored somewhere in this compartment, and then it should flow in this direction until it reaches the Hippocampus. So these are ducts until it reaches the Hippocampus. So we have the basal lamina, a form of connective tissue, or a protective layer around these acinas. It's called a basal lamina. If you need more information, histology books, they have a lot of information. And then we have the myoepithelial cells around the acinas. These are muscular tissues, just from the word myo, meaning they are muscles. Structural and functional unit of salivary gland. Myoepithelial cells, they are made up of muscles. These are the structures which are innervated. When we say parasympathetic, sympathetic innervation, they form neuromuscular junctions with the myoepithelial cells. The nervous system, they form synapses with these muscles. Myoepithelial cells, they are modified, or they are smooth muscles. Yes. So when the nervous system wants to regulate saliva production, they innervate these myoepithelial cells. And then we have the acina cells. Down here there's acinas. Let me show you acinas. These acinas. Acinas is the plural for acina cells. A group of acina cells, they make acinas. I'm emphasizing because it's the same with the structure of the pancreas and many other glands that you're going to face in the human body. Usually the structure is the same. So the acinas, it means a collection of acina cells. Okay. So acina cells, they can be in each acinas, you can expect about 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 to 12. Acina cells are, they make up the acina. Acinas, I mean. Okay, so these are the actual cells that are involved in the production and secretion of saliva. The acina cells. They are involved in the actual production and the secretion and the secretions of saliva into this space. What other structures do we see here? We see the intercalated duct. Intercalated duct, this is an area of high mitotic division, right? Mitosis. It's very high. It's an area where there's increased proliferation of cells here. Okay, these intercalated duct, they can replace the acina cells when the acina cells, they are worn out. So they are able to replace the acina cells, they are able to replace the striated duct. Striated duct, their function is the modification of saliva. To modify it so that it can become hypotonic. Because plasma is isotonic, right? So we want to modify the isotonic plasma into an hypotonic saliva. So that's a function of the striated duct. They are involved in modification. This is where you're going to see a lot of reabsorption taking place. We'll come to that. But I just want to emphasize the functions of these structures. Basolamina, just a connective tissue around or protective layer around the acinus. The myoepithelial cells, they are smooth muscles. Just like any other muscles in the body, they can contract. When they contract, they can squeeze the release of saliva. Okay? They are under the influence of the parasympathetic and the sympathetic nervous system. That is the myoepithelial cells. Even in the breast, you are going to find myoepithelial cells. When you look at the mammary glands, they also have this kind of arrangement. Okay, the mammary glands. You've heard of mammary glands? The lobules. Okay, they have this kind of arrangement in myoepithelial cells. Which topic do you do? Breast physiology or under reproduction? We'll do it. It's important to understand the physiology, the reproductive system. Very important. Acinar cells, I said these are involved in the actual production of saliva. The acinus is a combination of acinar cells. Interconnected duct, it's an area of high mitotic division. Striated duct, this is an area where there is modification of the saliva. So how is saliva produced? If you're asked in an exam, explain the steps involved in the production of saliva. So that when a patient comes in a hospital, has no saliva production at all, you should be able to remember the physiology. What do we call that condition where there is no saliva production? It's called if you know the function of saliva, you can predict what will be the outcome if in people with xerostomia. You can predict infections will increase. Okay, like fungal infection. Specifically, which fungal infects the mouth? Candida albicans. Thrush. I'm sure you've seen babies with, especially the newborn, maybe 3 months, 4 months, with the tongue whitish substances there. Right? That is Candida albicans. And also speech. Speech will be impaired, swallowing will be impaired. So these are not good conditions. So it is important to know the physiology of saliva production. It is formed in 2 steps. Some of you are going for a lab. Okay, we have 30 minutes. The first step is the formation of an acetonic plasma-like solution. The second step is modification of this plasma-like solution. That's all. Only 2 steps. The first step, the acinar cells, they'll be secreting an isotonic plasma-like solution. Same composition with the plasma. And then the second step, they'll be modifying that plasma-like solution by the ductal cell so that it becomes isotonic. Okay, now that's not enough. This is just based on transport mechanisms. Transport across the cell membrane. Active transport, secondary active transport. So as you can see, mechanism of saliva secretion. Initial saliva is produced by acinar cells and subsequently modified by ductal epithelial cells. So this is our acinar cells. So the first step, there will be release of isotonic saliva. So as the saliva is flowing, it will move through this lumen. As it passes through the ductal cells, the ductal cells will begin the reabsorption. They will reabsorb a lot of sodium and chloride. At the same time, they will be secreting potassium and bicarbonate. So saliva is rich in potassium, it is rich in bicarbonate. When you compare it with plasma. Okay. So there's questions in multiple choice question, multiple choice exams. Comparing saliva with plasma, which of the following electrolyte is abundant in saliva? I'm sure you've seen those in your past papers. Yes. Yes. We put potassium, bicarbonate, sodium, and you should be able to know. So saliva, when you compare it to plasma, saliva has more potassium, it has more bicarbonate. When you compare saliva to plasma, saliva has less sodium and less chloride. So this is the actual mechanism happening in the ductal cell when you isolate the one ductal cell. Like sodium, how is it reabsorbed? The mechanism is the sodium hydrogen exchangers. This explains why there is slightly acidity, because there is a hydrogen ions which are released into the lumen. What about the chlorides? How are they reabsorbed? By the chloride bicarbonate exchangers. What about the potassium? How are they secreted from the ductal cells? By the potassium hydrogen exchangers. Or transporters, if you want to call it that way. Don't get confused. Transporters, exchangers, or pumps. But they are all secondary active transport. And they are dependent on this common pump, which is called what? Yes, the sodium potassium pump. Any questions? So just first to the isotonic. We won't go into detail how do the acinar cells produce, how do they convert the plasma into isotonic? Not necessarily certain things. Unless you want to go into academics, when you specialize in what you call doctors that specialize in to do with saliva. I think those that gastro. like that. They are specialized to do with the digestive system. So those gastroenterologists. Specifically, they look at the digestive system. For 4 years. Specialized. So avoid failing, right? Failing. 4 times you're just failing. Make progress. Because when you finish, you have to specialize. Another 4 years. Actually, you are all directly into medicine. Okay, they are nothing like making points. Hey, wait a minute. How are things? What has happened? When you are in first year, you do the same things with those who are going to engineering?
No.
Okay, first semester, same things. LS. Second semester, you drop the physics. Oh. So, those who came in first year, they knew what they are going to do. That they are coming to study medicine. They knew. They knew that they'll study pharmacy. Wow, it wasn't like that. In our times, we used to make points. When you don't make points, you find yourself doing certain things you didn't want to do. Do certain things. I'm sure you know those stories, right? You find yourself doing certain things that you didn't want to do. But you don't regret anyway. The government was sponsoring you, and you realize that you just thank God whatever happens. Yeah? Because if you are in the village, but the government has decided to sponsor you, whatever you come to do, whether it's entomology, if you have come to do agriculture, even if you didn't want to do it, just do it. And biological sciences, just do. That's how it was in our time. But now, you know directly that, okay, I'll do this and this. That's how it is. Nothing like making points. So even if you get a C, you're still going to make that's nice. But who's that got a problem, because it would be, let's say, 1,000 NS, and then they just want 50 to go to medical school. Now capacity by Hippocampus, because the same capacity. Where have they where have they gotten the capacity? Now they were saying no, there's no capacity. Just 150. And they are what? Even now. That's why I'm wondering. They were saying just 50. 60. That's why there's been shortages of doctors. Somebody was stepping on, let me not go into, but somebody was stepping on people's careers. Jealous. Long time, je? Ah. Mari kwenda kwa soma guys. I don't want people to become doctors. But, nevertheless, life continues. Okay, so this is the mechanism of saliva secretion. While political, so orange the report. These are the students are very political. You are recording me.
That is for class. Come closer. I can't hear anything. I can't hear.
Come better well. You mean there are people that enter directly into bed? Yes. Oh. Which other courses were there? Microbiology and fluid state. Acina cells secrete the initial saliva, which is, okay, this I've already mentioned. Pardon? Now, the most important thing, regulation of salivary secretion. The best, this diagram can at least explain what I'm trying to explain. This is your acina cells or ductal cells. They have receptors, muscarinic receptors. They also have beta adrenergic receptors. So the beta adrenergic receptors, they respond to norepinephrine. The muscarinic receptors, they respond to acetylcholine. But when you give atropine, it can inhibit the action of the acetylcholine on the muscarinic receptors. Muscarinic receptors. The spelling is like this. Receptors are very important, but it's another topic on this one. And adrenoceptors. Especially when you do your pharmacology, it's very important to know the various types of muscarinic receptors and nicotinic receptors. There are various subunits. Since are proteins, you find there is alpha, beta, beta 1, beta 2. So what's happening here? What activates the parasympathetic? Conditioning, food, nausea, smell. When I just smell something, parasympathetic stimulation occurs. And then the innervation to the salivary glands, we have cranial nerve VII, which is 7. That is the facial nerve. And cranial nerve number IX, that is what? Glossopharyngeal. This is the innervation of the salivary glands. They release acetylcholine at their terminal end, activate the muscarinic receptors, activate these second messengers. We talk them under endocrine system. These are second messengers. The purpose of second messengers is just to amplify the acetylcholine, because these are released in minute quantities. So we need it to be amplified. So there is production of second messengers, IP3, which is inositol triphosphate, and the calcium. These are called second messengers. This is a first messenger. Second messengers, they are only localized in the cell, inside the cell. That's why they are called intracellular messengers. And then sympathetic, it originates from T1 to T3. You know what T1, T3 is, huh? Okay. It releases nitric oxide, epinephrine, activate the beta adrenergic receptors, resulting into production of this second messenger, cyclic AMP. So, what you need to know is the parasympathetic causes the release of watery saliva. The sympathetic causes the secretion of thick saliva. That's a difference. But they all stimulate the secretion of saliva. It's only that parasympathetic, watery, sympathetic, thick. Yes?
You said that the parasympathetic stimulated by smell impulse. What about the sympathetic? What stimulates it?
Very good question. The stimulus here, they have been shown here. Conditioning, we know you are doing psychology, is it? You know what conditioning is? Who can give an example of conditioning? From psychologists. Psychology. I don't know if it's you are doing psychology or sociology or behavioral science. But a good example of conditioning. Yes. Come again?
I'm saying a boy child is often given a reward after cleaning his room. He continues to clean his room despite him not being rewarded in a while. So that implies that he has been conditioned to clean his room. He always expects a reward. It's like variable ratio.
Yeah, that's condition. That's become a habitual kind of a behavior. Yeah, conditioning. So when you just pass through Hungry Lion, you start salivating. Just by seeing the logo for Hungry Lion, that's kind of conditioning. So, dehydration, fear, sleep, they inhibit. So what should, anything that activates the sympathetic system, like exercise, usually activity activates the sympathetic. You're following? When you are active, like running, doing some kind of physical activities, the sympathetic system, it is activated. So saliva secretion usually, it's a thick kind of saliva. So that's what we can say. Even anxiety also activates sympathetic nervous system. So just to answer your question, those are the factors that activate the sympathetic system. Okay. Otherwise, are there any other questions? So we'll finish the digestive system. So what you need to know is just to go through your slides. Okay, at this level, just get to know a lot of things about systems. If they are not making sense, And if they are not making sense, sometimes they'll make sense one day. You need to know the length of the digestive system. You need to know the diameter of the esophagus. You need to know the parietal cells, what they do. All those information. So that when somebody comes with a problem in your clinical practice, you'll be able to brainstorm. Huh? Because you have knowledge somewhere, you'll be able to relate certain information. So as physiology, when we prepare an exam, we are going to ask basic physiology. Do you know this? Which neurotransmitter is released by the sympathetic? Such kind of questions. Which neurotransmitter is involved in peristalsis? So just know each and every concept. Yes? I saw a hand behind. So on bowel, gallbladder, just watch that, just to supplement your reading. I recorded something for you on bowel and the gallbladder. You saw it, huh? Yeah. According to the schedule showing, we're supposed to start. I think cardiovascular system, something like that. Okay, prepare to go for the lab. We'll end here.
Great.